Okay, our final speaker today is Dr. Stephen Kern from the Gates Foundation. He's currently the Senior Program Officer and Head of Quantitative Pharmacology. He has a background in both mechanical and bioengineering, and he came to Gates Foundation, I think from Novartis, where he was the Global Head of Pharmacological Modeling. And the title of his talk, Accelerating Global Health Innovations Towards Real Solutions in the Field.
This guy is the laser.
Okay.
You have to advance the slides with the
Okay.
arrows.
Great.
Thanks.
Okay. Good. Thank you all, and thanks for staying long enough. I have the distinct advantage of being the last speaker, which means I can say things relevant to all the other presentations and make references to them, hopefully correct. I have the disadvantage in that I'm the only thing keeping you from the wine and the beer and the snacks that are outside. So hopefully you'll find that this is interesting. So I've been really pleased with the talks that I've heard today. I think they've been very interesting, and they really have focused a lot on the science and the basic science. And I want to just highlight some aspects about accelerating towards the real solutions in the field, because ultimately that's what all of you are doing. You do have a motivation to understand the science, you have a passion for that basic science, but I think everyone also would like to see that passion turned into something that helps people, just like we saw in the previous presentation, who are stuck with endemic conditions. And so one of the key tenets at the foundation that we sort of have as one of our mottos is that every person deserves a chance to live a healthy and productive life, and that's a big part of what drives us on a regular basis. And I think we can feel pretty good that they're making some progress in global health, at least over the last 50 some odd years. So you can see that there's been a great decline in the under 5 child deaths worldwide, but still a long way to go, with 6.6 million deaths attributed in 2012. And when you look at what are some of the main conditions that are contributing to that, I think one of the morning's speakers, if not two of them, actually had mentioned that pneumonia, diarrhea, malaria, and also other infectious diseases are a big contributor. Here you see these three contributed to almost half of that 6.6 million. And the interesting thing is that there's an understanding, or at least a belief for many of us, that these are preventable deaths, right? Because we know we can treat pneumonia and clear it in individuals. We know we can do the same for malaria and diarrhea, yet we aren't to a large degree. And so that says that we have a long way to go in our innovation to get to global health solutions to help these patients out. So what is innovation in global health? I'm going to offer you the Gates Foundation perspective, and it's not the Gates Foundation perspective, so it's just one of many. But I think one of the things that you've heard a lot is some great innovation this morning in the basic science approaches and talks that we heard. I was really pleased that I think in all of the presentations there was something in those biological descriptions that highlighted a target that somebody asked a question about, might that be a druggable solution, right? So that the stimulation of this basic science is a big part of the innovation, but there's other parts to it as well, because we have to think about what's going to take your publications in Science and Nature and Cell and bring them to the patients in Uganda that Warner talked about before. So we really need to think about those other factors. One of those other factors is just things like brute force production. So since 2001, the manufacturers that are now supplying Gavi-funded vaccines have doubled. So there's double the capacity to produce vaccines to actually have solutions to bring to people in the real world. The additional children, therefore, immunized with the support from Gavi has been 370 million over this period of time. So that's a great increase. And the drop in cost of vaccines has been 36%, in this case to immunize a child with pentavalent pneumococcal and rotavirus vaccines. So these are all driven by innovations. They're not innovations that are going to get published in Science and Nature and Cell, but they're really important innovations if we want to treat the conditions in the populations that we want to serve. So in 2009, rotavirus vaccines were $7.50 a dose. If you were saying that that was your final target, that's really expensive, but nonetheless it was enough to get the product into place, because in 2012 that rotavirus vaccine dropped to $2.50 a dose. Again, through better innovation, increased manufacturer, competition, all of the factors that would help to reduce that price. And in 2015, it's anticipated that that dose will hit the target that we'd be striving for, which is $1 a dose. Okay, so that's not to say that we shouldn't fund something or be involved in a project when our initial estimate is going to be $7.50 a dose, but we have to continue to think about ways that we're going to innovate so that we can bring those solutions down to a cost that make them affordable for the people that need them. So at the Gates Foundation, what we focus on in global health are basically two things: identifying areas of greatest need and where we think we can have the greatest impact. Because as you heard today, there are many other funders and many other people who are having a great impact in the global health. We're not alone, and we want to make sure that the things we're doing are complementary and not redundant. One of the important aspects, and if any of you follow Bill Gates' annual letter, if you do, it'll be out next week in his next annual letter. But last year's letter was all about measurement. And so the foundation is very, very key on data and measurement and understanding magnitudes of the problems that we're working with, and then goals, really quantitative goals to go forward. So here you see a grid plot that's just basically an assessment of the under 5 burden of disease. This is from IHME at the University of Washington, Chris Murray's group, and there it helps us identify at least that left side of the curve. What are the main conditions that are impacting the people that we're trying to serve, and where should we put our energy and our effort? And then on top of that, we look at the solutions, either solutions that we're funding or solutions that exist, to identify where some of these conditions are being addressed. So here you see a plot that just shows on the x-axis what we call the probability of technical and regulatory success, and on the y-axis, the dollars per DALY averted. So basically, what does it cost us to bring some benefit to the populations that are served? On the far right side, you see two circles, an orange and a less orange circle, and those are benchmarks. Those are very well-known interventions that have had great effect. One is surgical male circumcision, which you can see is very inexpensive to deliver and has a very large technical success if it's used correctly. And the other is bed nets. Again, a little bit more expensive, but bed nets, when used, are incredibly effective. And those become benchmarks at which we can assess other things and other investments in our portfolio to say, where is it possible for us to have the greatest impact? Where can we make investments that can move the field further along? It's also helpful for identifying where there might be gaps in things where we could go further with our investments or help work with partners to fill some of those gaps. So one of the big things that's happened, as Dan mentioned at the introduction, so I came to the Gates Foundation from Novartis. And prior to when I was at Novartis, the head of global development there is Trevor Mundell. And Trevor Mundell now is head of global health at the Gates Foundation and was largely responsible for my switching over to Seattle. And one of the things we wanted to focus on, or he's bringing as a focus to the foundation, is this fundamental challenge of how we can shift our development efforts to the left, in his words, for maximal impact. So here you see a curve that shows just the standard current paradigm that you would see for drug development. And that is that in the beginning, early stages, you have very low probability of technical and regulatory success, and also consequently generally very low costs. And it isn't until you are at these end critical stages, when you're enrolling lots of patients, thousands of patients in big trials, very complex to carry out in geographies all around the world, that you start really showing a shift in that probability of success and subsequently also a cost. Well, the problem with this is that it puts a big risk of failure for things, because you're going into a stage where you're going to spend a lot of money on a gamble that may or may not pay off. And so one of the big efforts we've been trying to focus on in the foundation in the last few years is this, what we call shifting the curve to the left. And that is by increasing the investment in the earlier stages, understanding more in the proof of concepts, getting to a characteristic so we have a mechanistic understanding of why drugs are working, and we're studying them in a way that makes detailed sense. We can hopefully shift this probability of success curve to the left. So earlier we have a greater estimate or guess that our success is going to be achieved. And when we enroll then into these big studies that are in phase 3 in large populations that take a lot of time and effort, our chances of succeeding are already relatively high. That's the ideal that we'd like to get there, or get to. And we're not alone. The drug development world would like to do that as well too. It's a tall order, but we think that the more we can put an effort, particularly into these earlier stage from preclinical to proof of concept components, and by increasing our investment there, we can learn more and understand what's likely to be successful, what's likely not to be successful, and to put our energy and effort into those that are. And then if we can do that, this might be a better representation of the future for development of drugs, vaccines, and diagnostics in the global health paradigm. So let me show you an example that I took with permission from my colleagues at MMV, so Medicines for Malaria Venture. So they have been working on developing new antimalarial drugs. We have good antimalarial drugs now in artemisinin combination therapy. But of course, as we heard earlier today, the emergence of resistance is looming. It's already been documented in Southeast Asia and hopefully won't be going further than that. So MMV has a suite of drugs in their pipeline that they are working on moving forward that have different characteristics. But this one, OZ439, is a new class of drugs not been studied yet or tested yet actually in malaria therapy. And so the specification was to create a compound that is capable of killing greater than 10 to the 6 plasmodium parasites within 96 hours. So when they got to the point of the development of this compound, they had just completed what we call a proof of concept study. They had ranged the number of doses from 200 to 1200 milligrams, and they had shown that it exhibited maximum efficacy over 36 hours. But at this stage, if you've got a brand new drug that you've never tested before in patients that have malaria, the way you have to study it is you give the drug for 3 days, and then by day 7, you have to switch them over to a known therapy that does work. Because you don't know if you've cured their malaria. You're not going to likely be able to keep them housed in a hospital for 28 days or 42 days to make sure that they don't recrudesce. And so the only way you can study this is to give the initial doses, look at the decline in the parasitemia, and then follow up at day 7. But you've lost a lot of important information about how effective those doses are at that stage. And so then you have to use this information to design what's called a dose range finding study that oftentimes has many cohorts. Here you can see they were planning on 12, involves a number of people. And at this stage, you don't have a strong link between the efficacy and the optimal dose. And so what they wanted to do at this point was to try to look at all the data that's been collected in the development of this compound and other antimalarials to see if they could get a better estimate of what might be the effective doses going forward, and in doing so make a more efficient trial, reduce the number of patients that have to be exposed, and move faster to their next stage. So they went back and looked at the data, and fortunately one of the biggest, let's say, innovative developments that have come forward in malaria is the development of the SCID mouse as a model, animal model for malaria. In the past, the mouse model for malaria was always infected with a strain of malaria called P. burgii. It's not the same as P. falciparum. It responds differently to the drugs than P. falciparum, and it's very hard to translate P. burgii IC50s to P. falciparum IC50s in humans. The scientists at GSK, at the Trace Cantos facility, have been able to develop a SCID mouse that they then can infect with human plasmodium. And now when you look at the exposure-response relationship of this animal model, you get a much better estimate of what's likely to be the effective dose in a human, because you can get a sense of the concentration effect response. So using that data, they then simulated what they thought would be the PK to PD results in the mouse, and the dots are the measurements, the blue lines are the simulations, and did a reasonable job. These are with very small numbers of mice, n equal 1 per dose, but at least giving us some idea of the relationship between dose and exposure in an animal model. Then that model can be essentially scaled to humans using very standard scaling routines across allometric routines, and give you some estimate at least of what might likely be the human PD response and the concentration you need to achieve. And again, this was done in collaboration with GSK. Then that gets translated from preclinical data to give an estimate of what we think would be the PD response in humans. So here you can see one set of results for another antimalarial drug that MMV had already done this characterization in, mefloquine. And so you see here on the left side parasitemia over time for a number of different doses, and then here on the right side parasitemia over time predicted. These are the data that are coming it's the combined model plus the observed data. And here on the left is what would be anticipated to happen in the SCID mouse when they went forward based on the modeling predictions. And you can see the simulation results and the variability in the simulation results and the actual measured data, all essentially giving you some estimate or some confidence, I should say, that you have a system where you can scale from the preclinical data to the clinical data and use it effectively. And then the key component to this that really helps it out is the creation or the development, again, an innovation in biomedical science that James McCarthy in Queensland is doing, which is a human challenge model to malaria. So this is giving subclinical doses of malaria to human volunteers who would like to volunteer for this study. You basically are given a small transfusion of cells that are infected from malaria. It's a population, a pool of cells that came out of the UK that have been characterized and used. And James's lab induces small, low levels of malaria in volunteers that then mature over 7 days and are administered drugs and tested for clearing. And the interesting thing that helps make this work is that by giving low levels of parasitemia to patients, you can scale them because they linearly scale to the large clinical levels that you see out in the field. And this time, or this slope here, this time that the parasitemia declines, is more or less parallel to what you see in clinical doses. So you don't have a saturation, you have a direct scalable response. So given that, now we have something where we can test the drug in an otherwise healthy but controlled population, get a sense for what is the right dose that's likely going to clear the parasitemia, estimate from the pharmacokinetic scaling and from dosing in human volunteers who are healthy without malaria, the true pharmacokinetics, so we know a dose of a certain amount will last over a certain time above the minimum parasitemia concentration. And based on what we see in the parasite model, estimate what that capability might be in people who have clinical loads. Now when I want to go into the trial in Thailand or in sub-Saharan Africa or in places where malaria exists, I'm much more confident that I have a dose that's going to be effective for patients. I can clear them quickly and I can move forward in understanding how to combine drugs for malaria treatment. And so here is basically the results that they did for their compound, OZ439. So here you can see parasitemia levels following mefloquine just as a reference, and then here's OZ439 that they had used before for doing the scaling, and then here's the predicted and observed parasitemia for one particular dose, 200 milligrams, and they had predicted that this dose would not be large enough and you would see some recrudescence that occurs after about 2 days. And in fact, the dots in the variant show you what the results were, and the dotted lines are the predictions. So this early learning was very helpful, along with the population pharmacokinetic model, to be able to use the challenge data in an effective way to identify a dose going forward. Then subsequently, built on a library of other drugs that have been put through this same series of models, SCID mouse, human volunteer, human challenge model, we can then combine these pieces of information to figure out what would be an effective dose of one compound matched with the second, because malaria therapy is always given at least with 2 drugs, again for combining resistance, and also because they have different characteristics from a kinetic standpoint. So here you see then the simulated pharmacokinetics, and an important characteristic, this minimum parasitemia clearance concentration that's shown for the 2 drugs, here in this case DHA, so dihydroxyartemisinin, and in this case the blue OZ439. And so this was their estimate of an effective therapy that would be able to clear parasitemia, stay above the minimum parasite clearance for the long-lasting drug, at least out to 16 days. And then this was then the basis for their selection of doses going into a larger clinical trial, a phase 2b trial. So identifying this divine dose for OZ439, they were able to identify that 500 milligrams was going to be effective as the combination. When they gave that in the first study that they had done, 4 out of 8 subjects did not have any recurrence by day 14. Then the other half did, which was predicted actually out of the model. The human challenge model combined with the PK/PD modeling helped them understand the properties of OZ439 by itself, and then combining it with DHA piperacin was able to develop a specific combination that would be effective and prevent this recrudescence that you get with Oz alone. But again, giving the longer lasting drug at an effective concentration so that it's not going to produce potential side effects by overdosing. So the impact of this development on time and cost was a savings of about 2 years and an estimated saving in cost of about $1.5 million, which is great in terms of the time. More important, the reduction in patients by greater than 40, so less patients that have to be exposed potentially to subtherapeutic concentrations and have to be, have to deal with the challenges there. There was a reduction overall in the cost, as we said, of the program. That's great because that means then they can direct that money towards other aspects of their development program. So again, as we heard today from a number of the presenters, the funds that we need to be able to carry these forward are limited, so we have to use them efficiently and effectively. And this is one way, at least in a translational approach, that we've been doing this in malaria and hopefully can extend in somewhat similar ways in other disease areas. Of course, we don't have challenge models in a lot of disease areas, so that's going to be a problem. But ultimately, the point is to try to link together the preclinical, clinical, and early clinical data in patients in a way to optimize the design of studies to produce effective doses for patients. And this just gives you the acknowledgement of the people that did this work. So let me go on then to just briefly talk about the product development network at the Gates Foundation. So the Gates Foundation funds a number of different entities, which you heard reference to today, called PDPs, so product development partners. And those PDPs work in all the different therapeutic areas. And we as a foundation then try to work with these partners and support them to help move their efforts further in a faster time zone. Because one of the things that we're working towards, if you look at the website for the foundation, Bill and Melinda Gates have used a term called impatient optimists. And if you've ever met and talked to them about projects, you'd find that they are truly impatient optimists. And so we are really trying to move the field forward. And one of our goals now is to deliver 3 to 5 new health interventions per year in the programs that we're funding. Those interventions are not scientific papers. Those are important and we support their development, but they're actually solutions, diagnostics, drugs, therapies that are working for the patients that we're trying to serve. So we've created this integrated network across what we call the value chain of the development for the global health space. It involves having products and the right products for the right patients at the right time. This again is the importance of things like going to Uganda to be able to see what is the condition on the field, because I can tell you, if you've not been to the places where where these solutions have to work, if you don't understand the limitations and the challenges that are there, you're very likely to be able to develop a solution that may have no impact whatsoever on the people that you're trying to serve. And that's not a good use of your energy, your intelligence, or your efforts as well. So you really want to understand the right product and the right patients and at the right time. You want to improve the visibility and the portfolio discipline, that is, by making sure that we're funding things that are meeting the qualifications and these needs, and integrate that into managing our portfolio effectively. To do that, we've had to increase our internal product development capabilities at the foundation, so we have more people like myself who have come out of industry. But for 15 years before I was in industry, I was an academic with NIH funding and ran labs. So I've seen both sides of the approach. And so one of the things that's been nice in the foundation is we have people who are accomplished scientists, but also have significant experience in drug development. And those have been helpful to help bring in the right kind of management for our portfolio. And then working with our partners to basically improve the performance of the output from our investments. And I think you heard Pony in the conversation today in the panel talk about the baton hand, right? So the baton hand of development is really key, because many of you are involved in these early discovery, maybe the preclinical phases, maybe you can even stretch into some of these other phases. These chevrons are by no means meant to be exclusive. But then from some point that innovation gets handed off to PD partners. You notice that pharma tends to back out, because at this stage, this is the risky stage. This is where things cost money money, the risk and the probability of success is perhaps not strong, and the pharmaceutical companies may not always have the interest. We even heard from the smaller ones today, they have funding and they have the ability to work in that space because they're getting grant funding to be able to do that, but they have a for-profit side that wouldn't take that risk otherwise. And so here's where the product development partners come in and have a big fill and can hand off. Once we have proof of concept developed, then we can perhaps bring in pharma partners for carrying off the bigger, larger studies that some of our product development partners can't do. Some of them can do it, and if they can, that's great as well. And then there are other parties that come into play. And it's important to recognize that we have to always be thinking about the regulatory authorities, because that's another characteristic we want to avoid. We want to not develop a product that is really tremendous, really groundbreaking, and we can't get it into people's hands because we don't have a stringent regulatory authority, WHO pre-qualification, or a country-level regulatory authority to sell the compound or administer the compound in that country. That is a big limitation that we've been working through as a foundation. We have a regulatory science group that actually works actively with the WHO and with country-level regulatory authorities to help improve that process. It's very challenging in a lot of ways. And so we as a foundation have now efforts in translational sciences. Our PSTs, so our program support teams, and the integrated development group that we have in the foundation that I'm part of, and integrated delivery. So we can also help smooth in this baton pass. We can be another runner in that relay race and help move along some of these products towards the final development. Here's just an evolving picture of our product development network. If your organization is not on this, I apologize. It's an evolving one. It grows all the time. I think every time we do it, we add new partners on here. But it's important to see that we've got product development partners, we've got country organizations, we have academic institutions, we have for-profit corporations, and we have to bring this together as a consortium of people that are going to move these solutions forward to have effective products for patients in the areas that we're serving. Without this network, it really is too big of a task for anyone to accomplish on their own. Within the global health division, we're separated along these disease areas, as I mentioned, which we call PSTs. And then we have these translational or functional organizations that work across all of these different disease areas. And the nice thing here is that now we afford ourselves the opportunity to get out of our silos, so that instead of just looking for a solution that's only for HIV, as we've heard, most of the places where we have HIV, we also have TB, we also have malaria. So how do we catalyze on the fact that we've got programs that are working in the same geographical areas with the same people who have 3 different disease conditions? If we deal with them as 3 separate entities, it's not very effective and not very efficient. And so one of the things that we've been developing is building up these cross-functional components so we can work effectively across these different disease teams. So as a global health group then as well, we not only focus on those development components on the disease end, but the delivery aspect as well. The delivery aspect is really key, and heading up delivery is Chris Elias, who used to be the president of PATH. And so Chris and Trevor work very well together to make sure that all of the conditions that we're trying to serve are well integrated to these other aspects in our development and delivery side, particularly with other disease conditions that are heading, for instance, polio, hopefully towards eradication, things like agricultural development, water sanitation and hygiene and nutrition, and then things even like agriculture and financial services, because all of this together is a solution for the problems that we're trying to achieve. If we look at this without considering all these pieces, then we're likely to not come up with effective solutions. So the integrated approach for global health is really key to our efforts. We're trying to combine a number of things together so that we can make sure that we have solutions that are going to work across the spectrum for the populations that we're trying to serve. And it's important also to recognize that not all populations are the same. So here again, global burden of disease estimates for 2 different regions. Here for, where's the Oh, there we go. Okay. Here for Central and South America, you can see the 1990 estimates of global burden of disease. Here for Sub-Saharan Africa. And in 20 years, a good amount of progress in Central and South America, where these boxes compared to what they were in 1990 have decreased, so the disease burden has gone down. In Africa, not so much. You can see that the gains are really quite modest, if at all. So it's important to recognize that there are multiple factors that are contributing to the disease, that those factors are not the same in every place that we work. And if we don't account for that variability and that difference, we're not likely to be able to succeed. So ultimately, we want to integrate all these pieces together and be able to put together programs that quickly and effectively provide solutions, working with all of you, academics, product development partners, company people, government agencies, to provide solutions so that we can achieve this mantra, which we have outside on the building. This is a picture of our visitor center. So with that, I thank you for your time. I didn't keep you too long from the wine and the beer, and I'd be happy to answer any questions.
Hi, thanks. I was wondering how diagnostics fits into that plan, the diagnostics products. Is that under diseases, or mostly what you talked about was drug development, I thought?
Yeah, I mean, I used the drug development examples, but the diagnostics, actually the diagnostics group within the foundation sits in the same group that I'm in integrated development. So we work across all of the disease conditions, and we have individuals who are specifically focused on diagnostics within a particular disease. So it's a key part of each disease strategy with individuals who serve the needs of that particular disease, but then a platform that's trying to work across all the diseases as well. And diagnostics is interesting because a lot of the regulatory challenges there are not quite as well defined as they are in say, the drug and the vaccine component, particularly when you get to pre-qualification and the ability for uptake on a country level. So that is still one of the issues that's requiring a lot of work, and WHO has recently sent some signals that they're moving towards that, in that direction. Yeah.
I was interested in the graph that showed the Gates Foundation funding now more in the translational area. Since, in fact, discovery research is necessary to prime the pump for translation, and since the NIH is not an adequate funding agency now, do you see any possibility of the Gates Foundation moving to funding more fundamental science in the areas of emphasis that the foundation has identified?
Yeah, what I see happening within the foundation now is that there is and I don't know what the percentage is. If I looked at Discovery's budget relative to, say, the therapeutic areas program that's going to be more into the translational development phases, I don't know what the relative ratio of those are. But one of the things that we are doing in the discovery space is funding basic ideas where we're trying to at least telescope where they're going to fit into the translational component of it. So funding a basic idea for the sake of understanding or learning has not fallen within our strategy. We kind of feel so far that that has been the uptake of others. But as you say, NIH and other funders are starting to drop off. That might have to be an area that needs to be reconsidered. But we recognize we can't just try to create a pipeline that's waiting for things that don't come through the pipeline. So there is always a balance there depending on what we see the needs are.
Could you describe a little bit about the probability of technical and regulatory success, what that process might be like, sources of information, and how often you go through that process as an organization?
Yeah, we'd like to go through that process on at least an annual basis. We're probably just kind of starting to get up to that speed. So it's fairly soft in a lot of ways, as you can imagine, because especially if you're working on a new idea or a new technology that's never been tried before, what are you going to benchmark it against? So generally what we'll try to do is benchmark against other things historically that we've seen to go forward. What are the characteristics in terms of data or evidence or support we need to be able to help a project move from one stage to the next stage in development? And then what we'll do is look at what is the capability of our investigators, our product development partners, the academic researchers that we're working with, to be able to gather that data currently, the way they're funded or structured, to be able to get to that message. Now, one of the things, or that next step, one of the things that might come out of that is a recognition that given the way this investigator or this team is funded, they're not likely to be able to get to that step. And if we increase the funding, that might increase the probability of being able to achieve that, right? So part of it is an assessment to say, what is it going to take to hit success? Because ultimately, that's what we'd like to do. We'd like to invest our resources in things that are going to be, successful and achieve. And it isn't just saying what's the least amount we can give. It might be what can we do to enhance the probability of success if we spent more money, if we got more information. And that's really what this shift to the left is supposed to imply. Earlier, we might invest more money in things than we would normally do because it's going to increase our ability to say, yes, this has a good chance to succeed, or it doesn't have a good chance to succeed. With Pony and Ulrich, we've been talking about some of the anti-secretories. We're doing this study right now in Dhaka in support with them, and as a partnership, it's moving along, but not at the pace we'd like to see. We're not sure we have exactly the right dose for this therapeutic at this stage, but that's part of what we're trying to identify in this study. So are there other things that we could do where we could gain some more information in parallel in the meantime that would help enhance our decision making? It's really to help support making smarter decisions for the next steps to move forward. Did I see? I will say one thing just quickly. There was this one comment in the previous session about what academic centers could do to help in this. And I think from my perspective, one thing I think would be extremely helpful is for the students in the audience and the people who want to get involved in the product development side, learn the language. Really important to learn the language of drug development, of device development, of diagnostic development, of the regulators, the way they think and the way they're trying to work. If you've ever been to a foreign country, you know it's really difficult to find the bathroom if you can't know the language. So know the language is really key. And understand the incentives. I think we heard a really good discussion of that in this session at 1 o'clock, where we said, what are the incentives of the different partners? People have different incentives, and it's important to recognize the for-profit incentives, the academic incentives to publish, and to figure out ways where it becomes a win-win for everyone. And then also teach those opportunities, maybe in collaboration with some of your other entities here at the school. So.
Well, that was amazing.
Thanks.
Well, we're not quite done. We have a reception outside. Don't go away. Keep mingling. We have a poster session for the first time. But before we leave, I want to first of all again thank all the speakers for their amazing talks and for your attention for the whole day. And don't go away. Let's keep socializing, and I'll see you here in a year, if not before.